Look at this diagram of the somatic reflex arc. When you touch a hot stove, the sensory signals from the receptors in your skin route directly to the spinal cord. They bypass the brain entirely. The handoff occurs right here at the relay neuron. The afferent sensory signal immediately transfers to an efferent motor neuron, pushing the command straight back down to the effector muscle. This reaction feels instantaneous, but neural impulses are not electrical currents moving through copper wire. Instead, they'd rely on biological wetware. To achieve transmission speeds exceeding 50 meters per second, the nervous system requires highly specialized microscopic engineering. A nerve impulse, or action potential, is actually a biological wave. It is driven by the physical movement of sodium and potassium ions back and forth across the cell membrane. Before a signal even fires, the neuron maintains a resting potential, a precise difference in electrical charge across the membrane, kept in equilibrium by the sodium-potassium pump. This diagram illustrates an unmyelinated axon. As sodium diffuses along the axolemma, the process is decremental. The signal physically loses strength as it travels down the fiber. To prevent the action potential from dying out completely, the axon must constantly trigger adjacent voltage-gated channels. This allows more sodium into the cell, regenerating the signal in a continuous step-by-step sequence. This continuous propagation relies on slow ion diffusion. It is far too slow for survival reflexes, demanding a structural adaptation to increase conduction velocity. The solution is myelin. This fatty, electrically insulative layer wraps tightly around the axon, increasing electrical resistance across the cell membrane and preventing current from leaking out into the extracellular fluid. Myelin forms discrete segments along the axon called internodes. Between these insulated segments are microscopic bare gaps known as the nodes of Ranvier. This chart compares the distribution of voltage-gated sodium channels. Along the myelinated internodes, density drops to roughly 25 channels per square micrometer. But at the bare nodes of Ranvier, that density spikes to nearly 12,000 channels packed into the same space. By insulating the axon and clustering the sodium channels, the architecture forces the action potential to skip the insulated segments. The electrical signal travels freely through the internode, then regenerates instantly when it hits the dense channel clusters at the next bare gap. This node-hopping mechanism is known as saltatory conduction. It bypasses the physical limits of continuous ion diffusion, achieving the high-speed transit required for a sudden reflex. While somatic reflexes utilize chemical synapses between neurons, tissues that require immediate, synchronized, and bidirectional responses, like cardiac and smooth muscle, rely on an alternative mechanism, the electrical synapse. This diagram shows an electrical synapse. Notice the cytoplasm of presynaptic and postsynaptic cells are directly connected through physical channels called gap junctions. Presynaptic sodium floods straight through these junctions into the adjacent cell, generating an action potential without relying on chemical messengers. In a traditional chemical synapse, transmission is delayed because neurotransmitters must physically cross the synaptic cleft and bind to receptors. Electrical synapses eliminate this step entirely. Electrical synapses solve the speed problem for localized clusters of cells. However, for high-speed, long-distance neural transit across the body, myelin remains the only biological solution. The insulating myelin infrastructure is not self-generating. It depends entirely on specialized supporting units known as glial cells to build and maintain it. In the peripheral nervous system, a single Schwann cell wraps itself around exactly one segment of an axon, forming a single internode. To put this into scale, a single sensory axon reaching from the human thigh to the spinal cord requires up to 500 individual Schwann cells placed continuously along its length. The central nervous system utilizes a different architecture. Here, a single oligodendrocyte extends multiple branch-like arms to envelop up to 30 distinct axonal segments simultaneously. These expansive glial networks and the axons they insulate function as deeply interdependent units. Sustaining this precise architecture requires immense, uninterrupted metabolic resources. Synthesizing and maintaining thousands of concentric myelin layers requires vast amounts of metabolic energy, primarily in the form of ATP. To facilitate this intense ATP production and cholesterol synthesis, oligodendrocytes depend on exceptionally high concentrations of intracellular iron. This diagram of an oligodendrocyte's interior illustrates a chemical vulnerability. Iron efficiently drives energy production, but occasionally interacts with oxygen, generating volatile free radicals. During physiological stress like hypoxia, iron homeostasis breaks down, triggering a massive release of free radicals that actively destroy the cell. The exact metabolic catalyst required to build the brain's high-speed network also introduces an inherent mechanism for toxic self-destruction. This specific oxidative damage drives the selective death of oligodendrocytes observed in demyelinating conditions, such as multiple sclerosis. Without the protective glial cell, the myelin sheath breaks down. As the action potential reaches the stripped segment, electrical current dissipates into the extracellular fluid. Saltatory conduction fails. The highly optimized nerve impulses degrade to the speed of basic ion diffusion, severing instantaneous neural pathways and producing severe motor and cognitive deficits. Clinical researchers are now exploring targeted iron chelation. This therapeutic approach aims to safely extract excess volatile iron from the brain to protect the surviving oligodendrocytes from oxidative stress. The speed of human movement is a massive feat of biological engineering, relying on thousands of miles of specialized tissue. Yet it is balanced precariously on the microscopic management of a single, highly reactive metal.